Hello, welcome back. So, we are talking about some sensors, and as time passes on, we should be now smart enough to understand these sensors in a different way. So, very basic thing what we require, because all life processes are dependent on O2, but you have seen that the measurement of this O2 in anything, any solution, even if in that environment also, only thing that you have to have some saturated solution. That means you bubble oxygen to some solution and you put that solution for your electrochemical testing or electrochemical analysis or electrochemical sensing of your O2 molecule. So, this is very important and a very straight cut one. Now, we slowly move to something where we can define it as a biological thing, how biology can benefit all these things, and also the medicine or the medical people can also get benefit from this sort of measurements, because we are linking this and this O2 molecule, everybody should know that thing, a chemist, a biologist, a doctor, and everybody. It is talking, we are talking in terms of the corresponding O2 present in blood, in any kind of fluid what you have in your body. That means the dissolved oxygen is always very important. But in terms of something, that means if you go to a medical doctor, that if you just go for your blood to be tested, and this blood can also be tested for the presence of your O2 molecule, that means the dissolved O2 in blood. That is why I told you that how your myoglobin molecules, MB or the hemoglobin molecules are involved, because this we know that this can bind to O2 molecule and hemoglobin can bind 4 O2 molecule to give you species like this. So, these are very important and these are also interrelated to know. Now, another component. So, anything which can be analyte of interest for this thing, that means if we talk in terms of the corresponding development of these sensors, and we are talking about electrochemistry, the very basic thing what we are talking from our school days also, that means electron transfer, electrodes and all things. That is why we put electro. Now, we are bringing something, we will take out something from this biological part and we put here, giving rise to the study for bioelectrochemistry. So, not only your materials, solid materials, inorganic complex or organic molecules, you can detect its presence as well as quantify, because analytical chemist's job is how to find out the amount present in it. That means qualitatively you detect first whether oxygen is present, or quantitatively you find out the amount present. So, the same blood if we can handle, because we know that a common disease we are facing, the aged people also, the diabetes. So, the presence of glucose in the blood, we can detect. Why we are talking all this? Because it is not in that particular one, bioelectrochemistry, for whom the doctors, the pathologists whom are responsible for making these measurements or quantifying the amount of all these species. So, they are looking for something we call clinical chemistry or clinical analytical chemistry, which is very important, the clinical analytical chemistry we should know. So, if you have the glucose, and if to a chemist, particularly to an organic chemist, to an organic chemist, what the glucose is a typical molecule, it can come from a dextrose, it can come from other type of species, maltose, fructose, everything. So, is a sugar molecule. So, patients who are suffering from diabetes, they are having, we call as a sugar content or the sugar control of that. So, we determine the presence of glucose, say in 100 milliliter of blood, how much glucose is present. So, glucose to us, to any organic chemist, is a molecule which is organic molecule, and to an electrochemist Why we are looking for this glucose for this determination? What we should know, because the sensor is electrochemical sensor. So, your electrochemical sensor should sense the electron transfer, and we know that this is a nice molecule which can sense. Now, what you have to see? You have to see 2 or 3 things together, that whether this glucose molecule is electroactive or not. That may not be so easy to understand, because most of these organic molecules we know if it goes for oxidation, you may not get the corresponding oxidized form like we get for the metal ion. The ferrous ion is oxidized to ferric ion. We can have the sufficient stability for ferrous ion as well as the ferric ion. But if it is a glucose molecule, whether that glucose molecule is oxidizing to give you a oxidized form, which is also sufficiently stable in terms of only electron transfer. But to organic chemists, we all know that if they have some species like RH, they are happy to talk in terms of this formation as ROH, or if some carbon is coming out over here, you can consider that means R prime COOH formation. That means the carbon center having this, that means this a carbon hydrogen bond can go to carbon OH bond, and the other carbon also, if it has something, that means it is going from a alcoholic part OH or to aldehyde part CHO or a carboxylic part. So, what we are looking for here is that we are doing nothing, but simply we are rationalizing all these things. How you look at the problem, so is defining the problem to choose the problem also, that your O2 is coming, that whether you get this O2 from a cylinder, that oxygen cylinder, or whether you get it from air only, that O2 is there and O2 is being inserted. And sometimes if it is not coming from the air, we say that no sir, I have given some reagent for this organic transformation. So, it is coming from some organic reagent also. So, we know that a very good reagent for transferring those oxygen is your peroxides, so organic peroxides, perbenzoic acids and all these. So, the reagent, but how these reagents are we are making, that reagent can also be making directly from this O2, and there are some other molecules also like this myoglobin, hemoglobin and hemocyanin, we know they also bind oxygen. So, binding oxygen is something, and this oxygen utilization for your oxidation to convert RH to RH is something different. So, if we can utilize this oxygen for glucose thing, that means this oxygen is being utilized for glucose, and glucose is your RH. You know that some other groups are also present, that means that COHO is there, but the term you should know that we know that if glucose is oxidized, we get gluconic acid. This is also a organic acid. So, what does it mean? That means also that if you have something, some groups like alcoholic function or a carbonyl or carboxy function like aldehyde is there, which can be oxidized straight away to its corresponding acid. So, these two informations, whether we can develop some process such that we can go for your electrochemical measurements and bioelectrochemical measurements, and we can develop like your oxygen sensor, a glucose sensor. So, the problem is that how you can develop a glucose sensor. So, it should be now we bring something that your life is much more complicated, you believe that we are bringing something enzyme. So, you see a chemist should also know little bit about the enzyme, because we are should be expert very much, because we know that there are certain group of enzymes which can be defined as metalloenzymes, that means the metal ions are there. So, who should know best about those metal enzymes? It is the chemist, rather the inorganic chemist or the chemist who are dealing metal ions. So, metal ions you know very much, so why you should not know the corresponding enzymes or the metal enzymes? So, the sensors what you can develop for glucose is a enzyme based sensors. So, what we have seen for your case of O2 molecule, the thing is very simple, that you get this for direct measurement of this O2 for the sensor, but you have the electrochemical glucose sensor, and we can talk that is a organic molecule. If you consider that glucose, it can be a carbohydrate molecule, the carbohydrate chemists can also claim that it is a carbohydrate molecule. So, electrochemical behavior, and when you bring the glucose is the organic one, so organic electrochemistry you are doing, but when you bring the enzyme from the biology area, the field of biology is giving you the enzyme. So, we can directly consider this as a study on bioelectrochemistry. So, in different clinical laboratories, the pathological laboratories, they handling the clinical chemistry or clinical analytical chemistry for their routine day to day. It is not a analytical chemist job. Once it is standardized, it is the job of a chemical analyst. So, there are large number of these chemical analysts or the clinical analysts are there. So, they are handling all these things in routine way, that means 100 or 200 samples they can determine in a day. So, glucose in blood serum. So, you have to have the corresponding serum sample in hand and you take this. So, how you develop the thing? So, if you know nicely how oxygen sensor has been developed, you can develop the other one, that means the glucose sensor can be developed, because the basic principle or the basic idea behind the development of that sensor, indicating sensor, that is indicator electrode, so indicating sensor which can detect the presence of glucose in your blood sample. Now, what you have, because already we have discussed a lot about the membrane, the porous part is fabrication with respect to the electrode. In this case is more complex and consists of 3 layers now. Earlier we are talking about one particular layer and a porous part which is allowing the oxygen to enter. Now, you have an outer layer. So, it is not much complicated. What you go in a stepwise fashion, you are moving from one after another, that means you are going from one layer to the second layer to the third layer. Due to the introduction of this enzyme, because you have brought enzyme, where to keep that enzyme? So, we brought that particular enzyme and we have seen the earlier that how you can frame that thing, that you have the electrode, then some mediator you can have, then the enzyme you can have, then the substrate you can have. So, layer after layer you know already that how you can get that. So, now you bring the outer layer, now you see some polymeric material. Earlier we are talking about the polypropylene cloth, then the Teflon film. Now, we are bringing another thing which is also industrially very important, is your polycarbonate film. So, is the polymeric material of large number of carbonate groups only. So, the polycarbonate film, so the PC material is known as PC like PP, which is permeable, that means which allows to pass something, that means glucose is the bigger molecule compared to your O2. So, glucose, it allows the glucose only to pass, but is impermeable, that is the selectivity comes into play, that the size, the pore size, so is a semi permeable membrane, semi permeable in terms of allowance of the glucose to pass through that membrane, but it retains protein and other constituent of the blood. So, it is a typical filtration process which allows glucose to pass through that membrane. So, this is there for the outer layer, the polycarbonate film layer is there which allowing glucose to pass, and in the middle layer we will trap the enzyme, and the enzyme is immobilized because it is encased, it is trapped. That is why you get, write it as the immobilized enzyme. So, there is no mobilization of the enzyme part, because enzyme is the most costliest part, only for several cycles only, say 500 cycles or 100 cycles is a biologically originating catalyst, we all know. So, this enzyme should be immobilized on the electrode surface, trapping for the middle layer, and the enzyme is nothing but glucose oxidase is now what is therefore we are talking about the enzyme. What we are bringing is looking for some oxidation reaction in terms of oxidase, that means oxidase. Oxygen will be one more extra oxygen from the air will be inserted in the glucose molecule by this glucose oxidase itself. So, glucose oxidase reaction will take place. So, your electrode is there. So, who will sense the corresponding electron transfer or the current for this particular reaction? So, who will develop that particular current? Because we have seen that the formation of water molecule from O2 can be detected by electrode or the sensor. Now, you get one after another everything, that means the arrangement. So, this arrangement is basically giving us all these things in terms of the electrode you have, then you have the inner membrane. That inner membrane, as we discussed earlier, that you have a thin film of electrolyte. So, it basically is no direct contact with that electrode. So, is a very thin film of electrolyte you can have, then the middle layer and then the outer layer. So, this outer membrane, which is made up of polycarbonate membrane. So, the outer membrane is basically you have. So, this the whole thing is your outer membrane is rejecting the interference, that the protein part or the other species what can be present in the blood is basically the arrowhead is in the opposite direction, that means it is basically rejecting. So, when the beta D glucose, so one form basically what we basically find. So, this particular glucose, that means we consider that the glucose can also be attached to your hemoglobin molecules or the myoglobin molecules that we consider as the glycated hemoglobin. So, hemoglobin percentage we determine there also how glucose is attached to the hemoglobin molecules also. So, this is that beta D glucose molecule which is present in the blood. Why it is there in the blood? Definitely. So, that is why it is attached to the hemoglobin. So, this particular glucose, then since it is going through the semi-permeable outer membrane, it is passing through this, and your interferents are getting rejected. We do not allow the interferent, because otherwise everything will be their hodgepodge. So, you reach their GOX layer. GOX is nothing but your glucose oxidase. Enzyme is your glucose oxidase. So, enzyme is being trapped there. So, enzyme cannot move away. So, this can be considered as a typical enzyme electrode. So, your enzyme electrode will be responsible for showing your measurement for glucose, which we can consider as a subject under your bioelectrochemical measurements. So, when it is passing through your GOX layer and it is not directly reaching your electrode. So, nothing is reaching to your electrode, that beta D glucose is not reaching your electrode, it is reaching your enzyme area or enzyme layer or enzyme concentration or enzyme molecule, but we are writing something. That means, you see now your O2, that means what we are talking in our previous class, that O2 can be reduced by 2 electron reduction to hydrogen peroxide. And just now also in our previous class we have seen that rotating ring disc electrode. So, rotating ring disc electrode can have some sense for formation of your water molecule from O2 or formation of hydrogen peroxide from your O2. So, depending upon the nature of these reduction reactions, that we should exploit nicely to understand the path what is getting over there. So, when you have this, that means the enzyme what we are talking about. So, enzyme is your GOX, the glucose oxidase enzyme. So, this enzyme GOX is there, and what we are getting, we are looking for your O2 and we are going for this. So, enzyme GOX is there. So, O2 is therefore, so you have the glucose molecule forming to gluconic acid. So, what you get therefore, that the gluconic acid is forming, okay, fine, that is the fate of these oxygen. So, one of these oxygen can be inserted for your gluconic acid and the other oxygen molecule can give rise to the formation of H2O2. So, if the same amount of H2O2 is formed together with the formation of the gluconic acid, there is no need to detect this oxidized form of glucose, that means the gluconic acid. You can only detect this presence of this concentration of this H2O2, as we know that this is also electroactive, that hydrogen peroxide you can also detect by the electrode for its concentration. So, this H2O2 will now pass through your inner membrane, then again some selectivity comes into the picture, that your inner membrane can only allow your H2O2 to pass through to reach the electrode, and electrode is now will work on your H2O2. So, your chemical reactions in terms of the electron transfer, that means electrodes are now doing your chemical reactions or the chemical transformations. At the same time, your enzyme is doing its enzymatic function, working on your glucose molecules. So, large number of species of substrates you can think of in a fashion similar to this, that if you go for something, some oxidase reaction. So, a general terminology for this is that we can study basically several oxidases, these enzymes, these are your oxidases. So, is a oxygenation reaction, that means oxygen insertion reaction. So, oxygenation reaction on the gluconic acid. So, these oxidases are there. So, oxygenation reaction is the direct insertion of the oxygen on these oxidases. So, large number of oxidases you can study, where you have the substrate like this glucose to get the product, and indirectly what we will be looking for, the fate of your O2, this O2 is giving your H2O2. So, during that particular process, at how much H2O2 is forming, because everything is selective, no other reduction process is taking place on your O2 molecule, and this O2 molecule is giving only your hydrogen peroxide. So, we will measure hydrogen peroxide in this specific situation. So, these reactions we should see now. So, the inner layer, the last layer, as we have seen that it can be a simple electrolyte layer, but is made up of something, some polymeric thing, but is also a paper like thing, is a cellulose acetate membrane. The papers we all know that is a cellulose, polymeric cellulose. So, cellulose acetate is also a very fine film which is permeable to small molecules like H2O2. So, everywhere the modified electrodes are there. So, what we see that modified electrodes, it is the cover up of the electrode, but we are using some membranes, and these membranes we are talking about their permeability, their type of permeability. So, the type of permeability is such that some molecules can pass through the membrane and reach the modified electrode. So, when you have the cellulose acetate membrane, and we know it is selective for your hydrogen peroxide, it allows hydrogen peroxide to pass in. When immersed in a glucose containing solution, glucose diffuses through the outer membrane into the immobilized enzyme, giving this reaction. So, glucose plus O2 is giving rise to H2O2 and gluconic acid with respect to your COX, the glucose oxidase enzyme. So, glucose oxidase enzyme is working to convert glucose to gluconic acid and hydrogen peroxide. Then this hydrogen peroxide is next diffuses through the inner layer of membrane, and the inner layer of membrane is goes, that means the inner layer is the cellulose acetate membrane which allow your H2O2 to the electrode surface, and then your H2O2 is oxidized. So, the current as well as the required potential for the oxidation of hydrogen peroxide will be judged now, and you will be oxidizing your hydrogen peroxide back to O2. That means, it is not any kind of other disproportionation reaction or any kind of side reaction. That hydrogen peroxide can also give rise to water and hydroxides, also hydroxide ions, but this basically go for a oxidation reaction in presence of some OH minus. If the medium is not alkaline, it gets from the aqueous centers, that means the water molecules, to give back your O2, water and 2 electron. So, you get this, so that in one way is basically a sensing mechanism where we talk in terms of the corresponding O2. So, you now correlate these two sensors what we are talking from our previous class, that in one case we were having the O2 sensor, that oxygen electrode. Now, the product is your oxygen now. So, basically the previous one is the oxygen sensor or oxygen sensing electrode use, and this is an sensing mechanism or the sensor for hydrogen peroxide. So, this can also be used for any 100 reactions, enzymatic 100 reactions where hydrogen peroxide is produced. So, do not try to learn all this in a fashion that you will be learning only glucose, but it is not that. You have to apply this if you know the basic understanding that what is forming and how we are detecting electrochemically is the formation of hydrogen peroxide. So, if in the nature, in the laboratory, in the industry or in any other activity, if hydrogen peroxide is produced in the medium, you should be able to detect that and you can follow up why this hydrogen peroxide is forming. Even for in water also, something that somebody can tell you that due to water pollution we are getting some other obnoxious things, and together with that we are getting some amount of hydrogen peroxide. So, what thing you should do first? Okay, see, let us find out the hydrogen peroxide concentration over there. Then only you can try to find out that which is responsible for the generation of production of hydrogen peroxide in that water medium. So, you have to sense that. So, we are bringing so many things, that means enzyme we are bringing membranes, the different types of membranes, it is a part of the fabrication of the electrode. So, for fabrication of these electrodes or making all these things are very much related to the corresponding formation of this hydrogen peroxide detection. So, it is a sensor which is giving you the hydrogen peroxide. So, what you sense then, since the electrons are coming out from the hydrogen peroxide for giving you the oxygen and water molecule and 2 electrons. So, you measure the current. So, you measure the current. So, the resulting current which will be proportional to the glucose concentration of the analyte solution. So, what we are going for that? We were looking for the unknown concentration of this glucose. So, what should be your concentration? And any substrate, suppose industry is making your glucon D for us. So, if they also want to measure, because you require a very small sample, the amount of sample, because our next classes will discuss that how you make or how you get the real samples and how you prepare those real samples for analysis. So, this concentration is very less, and there also you can determine the typical what is glucose. So, this concentration is proportional to your current. You should remember it, the current is I value. So, this is the thing that when we talk in terms of the electrochemistry, always remember that either potential or the current now is a very simple way that you determine the corresponding current, the amount of current will be proportional to this, and as I told you earlier that whether you have this particular mechanism or any kind of amperometric measurements, you can go for a nano ampere level of current. So, whatever small concentration of glucose you can have, there is no requirement for your enrichment of the analyte concentration for glucose. You can directly use the blood sample for that. So, for the typical activity for the last 30 or 40 years, industry people are making these glucose sensors very nicely, having some disposable electrodes. Definitely for some measurements your electrode is working, otherwise you have to discard it. So, this nanoampere level, very low level of concentration of glucose can be determined, because you can detect nicely, you can amplify the current. So, the resulting current will be very much proportional to that of your glucose solution. Okay, thank you very much.